The outer membrane of mitochondria is completely permeable to the contents of the cytoplasm, while the inner membrane is extremely selective. Only certain substances can cross it, and they do this by using carrier proteins or special channels. The inner membrane is extremely folded, forming a series of deep invaginations called cristae. The inner mitochondrial space, or matrix, is where the Krebs cycle takes place. Pyruvate produced during glycolysis in the cytoplasm is broken down into carbon dioxide and water, with the production of NADH and FADH2. These two compounds supply electrons and protons that are subsequently used to produce ATP. Electron-transporting proteins are arranged in a specific order in the inner mitochondrial membrane, where they form the electron transport chain. The transfer of electrons along this chain is accompanied by the pumping of protons from the internal space of the mitochondrion into the intermembrane space. This creates an electrochemical gradient across the inner membrane, down which the protons return to the mitochondrial matrix. They do this through special channels associated with ATP synthase, which occurs in the form of stalked particles on the cristae of the inner membrane. The movement of the protons is coupled to the phosphorylation of ADP to produce ATP. Finally, the protons are bound with oxygen to produce water. The whole process is called oxidative phosphorylation. Mitochondria differ between organs. Those present in organs secreting steroids, for example the gonads and the suprarenal cortex, have tubular cristae, while those in organs producing proteins are characterized by flattened lamellar cristae. Apart from their role in energy production, the mitochondria are also efficient stores of calcium ions, for example in striated muscle fibers.